So hello everyone, welcome back to another particular lecture series of our Asymmetric Synthesis course, and this is lecture 4. And in our previous discussion we are talking about the generation of asymmetric catalyst followed by chiral pole synthesis. So in this particular class we are mainly focused on chiral auxiliaries and chiral reagents. Okay, so first, you have to understand that what is the chiral auxiliaries. Chiral auxiliaries is a chiral molecular unit which can be temporarily incorporated in a achiral substrate to guide selective formation of one possible enantiomer. Like in our chiral pole synthesis, if you remember, then their synthesis start from a chiral compound. Okay, and by different kind of functional group transformation we get our asymmetric product. But here the starting compound in the chiral auxiliaries, the starting compound is achiral, which a chiral unit has actually temporarily attached here to make this particular compound a pseudo chiral compound, pseudo-chiral. Now, depend upon this particular extra temporary substrate that is called the auxiliaries, this actually determine the stereochemistry of certain nucleophile, and whatever it is attaching here in this particular achiral moiety, that particular stereochemistry is influenced by this particular uh, chiral auxiliaries. And after that, uh, this is your auxiliaries and this might be your product. After that, this particular auxiliary part getting removed to get product having a chiral compound. Chiral product has been observed. That means in the chiral pole synthesis we had begun from the chiral molecule itself to getting another chiral molecule. Here we began with the achiral molecule, we'll temporarily introduce chirality here, and we do the functional group transformation and got the product, and after that the particular temporary chirality can be removed to form the ultimate our direct product. So working principle of chiral auxiliaries that I already discussed, that chiral auxiliary incorporates a specific steric hindrance upon formation of one particular transition state. Hence, the stereochemistry of the new chiral center can be rationalized based on auxiliary state conditions. Now this particular term we will understood with an example. Okay. So before that, there are certain criteria is there to form a good chiral auxiliary. Like any arbitrary chiral moiety will not serve as auxiliary. It had some kind of properties to make them suitable for acts as a chiral auxiliary. First is that it needs to available in both enantiomer form, because you don't know that in your final compound you need R or S. So as the chiral auxiliary influence the steric, so you have to be prepared. The chiral auxiliary should be in the both enantiomer form, that is R form, S form, both are required. Second is that need to be easy and quick to synthesize, obviously, because you don't have to put so much synthesis step to form a chiral auxiliary. Third one is the must be readily incorporated onto the chiral substrate. This is very important. Also should provide a good asymmetric induction, because in the transition state, if that particular chiral auxiliary doesn't provide a good asymmetric induction, then it might happen that the desired enantiometry excess was not observed. The fifth point is that should be selectively cleaved from the substrate under mild condition and must be recoverable and reusable. This is very important. So you can think about that chiral auxiliary is some kind of a protecting group, but which actually induce the chirality on the particular achiral substrate. So this particular question has been asked in every interview if you are giving PhD. So they will ask give an example of good chiral auxiliary. So remember, the good chiral auxiliary is Evans auxiliary, and this is actually used for like aldol condensation, also asymmetric aldol condensation, as well as the alkylation reaction also, that we'll discuss in the subsequent slides. So this is the structure. You can see here that S enantiomer and R enantiomer both are present here. Okay, so let us understand the synthesis pathway of this type of Evans auxiliaries. Basically, Evans auxiliary is actually derived from the natural amino acid. So this is the S-phenylglycine. This particular COH acidated part has been treated with BH3 or NaBH4I2 to form their corresponding alcohol. Followed by a C1 synthon has been used, where this both are actually leaving group. So it will just form this type of compound, and here this particular part you can see the stereo part. So this particular auxiliary has two particular center. The upper part are actually, this NH part are actually attached with the achiral part, and this phenyl group is actually having a stereo center, a chiral center here. This particular part actually determine the stereochemistry of the product, and this part actually attached with the achiral reactant. Okay, now let us take an example where chiral auxiliary has been used to form a asymmetric product. For example, you can find here, this is our Evans auxiliary. Now this is nothing but an acid chloride. Okay, now this to react to form this particular compound. Now initially this is our reactant. This doesn't have any kind of chiral center or any kind of chiral induction. Now after attaching this particular Evans auxiliary part, a new chiral center is here. Now we treated with LDA to form this particular intermediate, that are enolate intermediate. Now you can see there is a two possibility, cis enolate and trans enolate, both are possible, but if this particular enolate has been formed, it will have some kind of steric repulsion with this particular group. That's why this particular enolate has been favored. So as this particular enolate is favored, followed by a alkylation takes place. So, and you can find here, to maintain the stereochemistry or the steric repulsion, this particular attached chiral center now become the below the plane, because this particular Evans auxiliary is bulky group present in the above the plane. To eliminate the steric repulsion, this type of stereochemistry has been observed. So we can say that yes, this particular stereo center is actually directing or influence the product stereochemistry. Like if it is in a, if this particular group present in the backward side, then this particular alkane or this particular benzyl part are actually attached in the above position. This is the bold bond. And after that, how can you remove that particular auxiliary? The remove can be easily auxiliary removed by lithium hydroxide in presence of H2O2, which leads to give a chiral product, which is our targeted asymmetric product, and we get back our auxiliary. So this is the typical reaction of asymmetric enolate alkylation by using chiral auxiliary. Similar can be used for the aldol condensation also, that I will not discuss here, but the similar strategy can be performed in the aldol reactions. Now, what are the advantages, disadvantages of chiral auxiliaries? The advantage is it is a high diastereoselective, and another thing that here you can find here, that when this particular asymmetric induction takes place and the final product has been generated with auxiliary, it is formed a diastereomer which is easy to separate via column chromatography. Okay, because this particular stereo center hasn't changed, this might be possibility to form this product also, but if it is only an enantiomer, then it is very hard to separate. But since it is formed a diastereomer, so it is very easy to separate via column chromatography. So this is another advantages that is already discussed, that it is highly diastereoselective. Second is that chiral auxiliary can be recycled, that I already discussed, that it attached as well as it is easy to remove, and sense of new chiral configuration can be determined through the X-ray crystallography. Now, this particular chiral auxiliaries have also certain disadvantages, such as that it might happen that both enantiomer of a chiral auxiliary is not readily available. Usually one enantiomer is far expensive than others, because This type of chiral auxiliary has been prepared from mostly natural amino acids. So natural amino acids, the S amino acids are very easy to produce, but the R amino acids are very hard to produce. So the both in for getting the both enantiomer is a uh, expensive or less expensive things has been come into the picture. The second thing is that auxiliary need to synthesize. That means the synthetic step will increase while using auxiliaries, and it is a multi-step process. And you know that in multi-step process, the maintaining the proper amount of yield is very harder. And the thing is that stoichiometric chiral template required for synthesis the auxiliaries. So it is a upgradation, but also with certain limitation. Like for the chiral pool, the complete chiral compound has been transformed via functional group incorporation to get the product. Here the particular that the synthesis of chiral auxiliaries required a chiral pool synthesis, and followed by it actually serve as a auxiliaries. So this is also a kind of disadvantages. So now let us talk about chiral reagent, that is a upgradation of chiral auxiliaries. Chiral reagents are the reagent which have chirality and influence to give a affirmative enantiomer. This is an upgradation, and here is the example that when we treat this particular compound with sec butyl lithium and uh, benzophenone, then we got the racemic mixture here. But when we treat the same with a ligand system, that is a minus cephotine, then we got exclusively this particular product. This is not a racemic mixture, this is exclusive. And this is that particular chiral reagent. So basically what happened, that chiral reagent is actually influence the transition state such a way that one of the formation of one of the enantiomer is increased. So we'll not discuss about more chiral reagent, we more discuss about the chiral catalyst. So in the next class, so with this we have to end this particular class. In this particular class we talk about chiral auxiliaries, chiral reagents a bit very briefly, because that is not that much important for the exam point of view, but chiral catalyst is very important. So we'll discuss in the next class. So thank you for listening.